Good afternoon and also welcome to all. So today we are going to discuss about the moist heat method at the temperature range 100 degrees Celsius. Actually in this method mainly two types are involved. First one is boiling, second one is steaming. That is boiling at 100 degrees Celsius. And using this method, that is boiling at 100 degrees Celsius, it kills most vegetative bacteria and viruses immediately. And during boiling, the material should be immersed in water and boiled for 10 to 30 minutes. That is the procedure coming under boiling. And also we should check that the lid of the sterilizer should not be opened during this period, during the boiling period. And using this method, certain bacterial toxins such as staphylococcal enterotoxins, which are actually heat resistant, and also some bacterial spores, which are resistant to boiling, can survive. So this is the main disadvantage. Most of the spore forming forms, also enterotoxins, are not easily destroyed using this method. Next is the boiling at 100 degrees Celsius for 5 to 10 minutes. It does not ensure complete sterility. So this method is not much effective, but it can be used also. But it is enough to kill all non-sporing organisms, and also a few spore-forming bacilli can be destroyed using this method. For example, strains of certain spore forming forms, that is Clostridium. Only spore forming bacilli, only few of them can be destroyed using this method boiling, but the strains of this Clostridium tetani, it is capable of surviving boiling for almost 1 to 3 hours. So after this we can destroy, but usually during this 1 to 3 hours of continuous boiling, the spore forms of this Clostridium tetani usually exhibit resistance and it can't be destroyed. So, and this boiling method in the medicine as well as in the microbiology, this method is used when the absolute sterility is unwanted. So, it can be used in certain procedures, boiling, which can be recommended. So, when the absolute sterility is unwanted for pipettes, for example, measuring cylinders, rubber stoppers, instruments such as scalpels, forceps, scissors, metal or glass syringe, for this type of materials, we can use the method boiling. And because this scalpels on glass syringes does not withstand higher temperature, so this method can be usually recommended. And also, but this boiling is not recommended for the sterilizing the instruments used for surgical procedures, because there is the need of absolute sterility. So it should be regarded only as a means of disinfection. So this method is regarded as a means of disinfection. And also in this boiling procedure, hard water should not be used. And this so while hard water is used, the effective boiling technique doesn't take place. So we can promote the sterilization by the addition of 2% sodium bicarbonate to the water to convert this hard water. So one point which we have repeated, when the absolute sterility is not required, certain articles and glasswares can be disinfected by placing them in the boiling water for 10 to 20 minutes. This is the picture of boiling. So, normally this boiling water procedure, which can be used for contaminated dishes, bedding and bedpans, because these articles doesn't require either sterility or the destruction of spores. Only disinfection is necessary for these articles. At altitudes over 5,000 feet, the boiling time should be increased. Because why? Because above the altitude over 5,000 feet, the temperature will be very less. So the boiling time should be increased by 50%. Because the water boils at this altitude at a temperature of 95 degrees Celsius or below. Then next method is, so we have discussed boiling at 100 degrees Celsius. Next is applying steam at atmospheric pressure or 100 degrees Celsius. So in this method, an atmosphere of a free steam, it is used to sterilize culture media which can be decomposed if it is subjected to higher temperature. So for this type, we are using a Koch or Arnold steamer. The laboratory autoclaves can also be used for this purpose. And this method is quite inexpensive method. And the container and the medium are simultaneously sterilized. And the evaporation from the medium, it can be prevented, and the apparatus requires little or no attention. So it is very easy to perform. This is the picture of Koch or Arnold steamer. And in this method, it is a vertically metal cylinder. Look, vertically metal cylinder, and it has a removable lid having a small outlet for the steam escape, and its bottom, it contains water which is heated up. So the lid portion is uppermost lid portion, it is conical and it can be removed, and an outlet of steam is also required. Then it consists, actually the cabinet of this Arnold steamer is tinned copper cabinet, and the walls are suitably locked, and the lid is conical, which enable the drainage of condensed steam. So the condensed steam can be drained out, and a perforated tray. Inside this Arnold steamer, there is a tray bottom, which is fitted above the water level, which is perforated, and this may ensure that the materials are placed on this perforated tray surrounded by the medium. And above this water level, the articles are kept on the tray. The tray is kept above the water level. During this procedure of sterilizing, the frequent opening of the steamer should be minimized, because when the air is permitted inside the steamer, it ultimately interferes with the sterilization. When the water is started heating, the steam can be generated inside the sterilizer at a temperature of 100 degrees Celsius. And thus the articles which are placed above that perforated tray, they are exposed to this steam for a period of 90 minutes. So that is the procedure of Koch or Arnold steamer. It is a quite inexpensive method. Next we are discussing about tyndallization or intermittent sterilization. This method is also coming under the steam at 100 degrees Celsius. Tyndallization is actually, it is a process which can be dated from 19th century. For what purpose? For sterilizing substances, usually food. And this method was named after its inventor, scientist John Tyndall. That can be used to kill the heat-resistant endospores. So this method can also be used for killing the heat-resistant endospores. This method is considered old-fashioned method, but still it is occasionally used. And this method is simple and effective sterilization technique commonly used today. So this method, but nowadays we are commonly using autoclaving. That is heating the substance sterilized at 121 degrees Celsius. That means above 100 degrees Celsius for 15 minutes in a pressurized system. Certain materials, when we are exposing to this temperature, there is a chance of destruction of that culture media. In such conditions, we are using this method. If the autoclaving is not possible because of the lack of equipments or the need to sterilize something that will not withstand the higher temperature, unpressurized heating for a prolonged period at a temperature up to 100 degrees Celsius, that is without applying pressure, and is unpressurized heating, and it is for a prolonged period at a temperature 100 degrees Celsius. The boiling point of the water, that is 100 degrees Celsius, may be used for the sterilization. So this method is used in tyndallization. The heat will kill the bacterial cells, and the bacterial spores capable of later germination into bacterial cells may survive. So this tyndallization can be used to destroy the spores. And it consists of heating the substance to boiling point or just a little below the boiling point, and it is held for 15 minutes in 3 days in succession. After each heating, the resting period is allowed. So this may allow the spores that have survived to germinate. So when they are germinating from the endospores to the bacterial cells, these cells can be killed by the next day's heating. During the resting periods, the substance being sterilized is kept in a moist environment at a warm room temperature, conducive to germination of the spores. So this may help in the spore germination by keeping it at a moist environment during the resting periods. When the environment is favorable for the bacteria, it is conducive to the germination of the cells from spores. The spores do not form from the cells in this environment. And this method is quite effective in practice, but it is not considered totally reliable. Why? Some spores may survive and later germinate and multiply. So it is not often used today, but is used for sterilizing some things that cannot withstand pressurized heating, such as plant seeds. The media such as TCBS, which is used in the Vibrio cholerae, DCA and selenite F growth used against the Salmonella and Shigella, these are sterilized by steaming. Sugar and gelatin in the medium may get exposed to free steaming for 20 to 30 minutes for 3 successive days through this method. The success of the process, it primarily depends on what? Spore germination. Though generally adequate, this method sometimes may fail with the spores of anaerobes and thermophiles. That is the disadvantage of this method. So, these 3 medias are used in the tyndallization. So, intermittent heating for 3 consecutive days. So, these are all about the moist heat methods which we are doing at the temperature 100 degrees Celsius. That means mainly boiling and steaming. In the steaming, we have discussed Koch Arnold steamer and intermittent sterilization. In the next session, we can discuss the moist heat method at the temperature above 100 degrees Celsius. Thank you all.